There we go. All right, so guys, gals, this is the slide that we left off on yesterday. We were talking about medical imaging and how it's really nice compared to cutting somebody open to look on the inside of them. There's a lot fewer side effects. Some of the ways that we have, or that we can look at the inside of a human being, involve x-rays. Most of these methods are going to involve some kind of a waveform or a wave function, either a sound wave or some kind of electromagnetic radiation. X-rays and radio-opaque substances are two examples that involve wave function. So as we look at an X-ray is going to make dense tissues appear white. And then when we look at how frequent X-rays are, it's the most common. It's the cheapest, it's the quickest. I typically get an X-ray done on myself probably once every 18 months, usually from the dentist. When we look at radio-opaque substances, this is a way to look at tubes or soft structures on the inside of the body. These radio-opaque substances are usually going to be some kind of a radioactive isotope, oftentimes a radioactive barium inserted into a glucose molecule. So we have radioactive sugar that a patient drinks. And then as that radioactive substance moves through the body, we find that the parts of the body that have the most fluids and absorb that substance the quickest, or absorb sugars the quickest, are going to be lit up. We also have computed tomography, also known as a CT scan. This is the new school term for something known as a CAT scan. When we look at computed tomography, you could think of it as lots and lots of X-rays. So this is X-rays on steroids. When we look at the CT scan, it's lots of X-rays being done in succession so that you can generate a three-dimensional image of the inside of the body using X-rays overlaid with each other. Obviously, as we look at this process, there's a lot of processing power that goes into taking these two-dimensional slices and turning them into three-dimensional images, hence the phrase computed tomography. We also have magnetic resonance scanning, or MRIs. If any of you are chemistry buffs, sometimes this is referred to as nuclear magnetic resonance, or NMR. It's the exact same technology. And what we look at with MRIs is that we have strong magnetic fields that take all the electrons within a molecule or within an atom and point their spin states all the same direction. And then the magnetic field is turned off. And the moment that magnetic field is turned off, the electrons start oscillating up and down with their spin states. And as you have an electric particle, like an electron, moving, it generates radio waves. And those radio waves are picked up by the scanner and used to compute the density of typically hydrogen atoms within the human body. And we find that soft tissues have lots of water in them, and water is 2 hydrogens, 1 oxygen. So these MRIs work really well for looking at the structure of soft tissues that contain lots of hydrogen atoms and lots of water molecules with those hydrogen atoms. And then we also have PET scans, also known as positron emission tomography. PET scans look for metabolic activity. This is another example of giving the patient a radioactive cocktail. You know, I mean, why not? Let's just have them drink a bunch of nuclear isotopes that are giving off gamma rays. And in this case, this is beneficial for the patient. These radioactive isotopes are going to be absorbed by the parts of their body that have the highest metabolic activity. We have an empty seat right there. The sound is good now. Sounds good now? Yes, we're awesome. Thank you for stopping by. So parts of the body with really high metabolic activities usually include nervous tissue, skeletal muscle tissue, or anything that you're using. So here we have a PET scan of the brain, and we can see that as we look at the frontal cortex on the top of the screen. Let's try this pointer right here. Does that work? Nope, that doesn't work. The frontal cortex is lit up, but as we look at the occipital lobe in the back of the screen right here, it's bright red. This person has a lot of metabolic activity in their occipital lobe. There's another part of the human body that has tons of metabolic activity that's just growing and growing and growing, and that's a cancerous tumor. Tumors or metastatic cancers have very, very high metabolic activity, and a PET scan is one of the go-to ways to isolate or identify tumors within a patient's body, because those tumors will be bright red beacons that are just scattered throughout the body. Typically, you'll find them in places that shouldn't have really high metabolic activity. We also have sonography, formerly known as ultrasounds. Sonography is beneficial in that we avoid the electromagnetic radiation. We avoid the X-rays. And instead of using EM wave functions, we're going to use sound waves to gather information from the inside of the patient's body. Ultrasounds have progressed quite a bit over the last several decades. 20 years ago, ultrasounds were fuzzy and grainy, and you couldn't get really good images out of them. And now with ultrasounds, you can get fantastic images out of them. Speaking from experience, about 6 months ago when my wife and I were going to one of her prenatal appointments, we were able to see in real time a three-dimensional image of our little girl moving around inside of my wife's uterus before she was born. And it's really cool. The figure up on the screen shows you what that can look like. You can make out lots of facial features of structures or children before they're born. So, that's how we can look at the inside of the human body. Let's talk about other fields of anatomy and physiology. These are going to be subfields within the discipline of anatomy and physiology. One of those subfields includes gross anatomy. And when we say gross here, the common joke is that it's yucky, it's icky, because it's called gross anatomy. But that's an overused pun. In the technical sense, when we say gross anatomy, we're talking about big. It's big enough that you can see it with your naked eye. For those of you that are thinking of going into a graduate program, so I know we have a lot of pre-professionals in here, or KINS majors. Most of the KINS majors or pre-professional majors in here are going to take a 5-credit gross anatomy class as your first class in your anatomy or your graduate program. And you'll have a cadaver dissection where you cut apart a human body. Another field of anatomy and physiology, a subfield, is histology. Hist meaning cell, ology meaning the study of. Hist meaning tissue, ology meaning the study of. When we look at histology, that's going to be the study of tissues. And it's going to be the primary basis of weeks 3 and weeks 4 in lab. We're going to spend a lot of time on that histology, looking at lots and lots of microscope slides. We also have cytology. Cyto is a prefix meaning cell. Again, ology meaning study of. Cytology is the study of cells. So that's a subdivision or subfield within anatomy and physiology. And we're going to camp a lot on this slide. So let's talk about the general pattern. If we start from smallest to largest, smallest being at the bottom of the screen, we find that if you take the smallest things and you combine them, you can get the next largest thing. So let's think of atoms and molecules. If you take multiple atoms and combine them together, you can make a molecule. If you take multiple of whatever is at a certain level and combine them together, you get the next highest level of organization. So multiple atoms combined together give you molecules. Multiple molecules combined together will give you organelles. Multiple organelles can give you a cell. And we need to pause at the cell. I want you to circle the cell. The cell is the smallest living thing. This is a common exam question. The smallest unit of life is the cell. The smallest living thing is a cell. If it's smaller than a cell, it cannot be alive. So molecules are abiotic. They're non-living. If it's a cell, it's alive. And if it's larger than a cell, like you and me, we're alive, but we're bigger than cells. We're made of cells. So let's go back to cells. If we have multiple organelles working together, they're cells. Multiple cells working together gives you a tissue. Multiple tissues working together gives you an organ. Multiple organs working together give you an organ system. And we'll pause for a moment on organ system. When we think of an organ system, it's the digestive system, the respiratory system, the nervous system. Just under half of you have had lab already. So you've gotten to spend some time working with the torso models and looking at those organ systems. Hopefully you've had a chance to take apart the torso model and look at the organs of the various organ systems. If we have multiple organ systems working together, we get an organism. And in this class, the organism we care the most about is a human being. So, let's get a little bit of feedback from you. Let's take a moment and fill in the blank. A blank is a compound composed of 2 or more tissue types, whereas blank are microscopic structures inside of a cell or in a cell. And just to reiterate, I've gotten a lot of emails about this. For these in-class clicker questions, I'm not going to penalize you if you answer them wrong. I just want participation from you as we're working through these clicker questions. So make sure that you try to answer them.
Notice at the beginning of this, it says that you're recording these lectures. How do we do that on our...
Ooh, how are you going to review the lectures? I'm going to post the recordings on YouTube and then put a YouTube link on Canvas next to the PowerPoint that it corresponds to. Good question. So let's go 10 more seconds. If you haven't answered yet, please make sure you answer. We have 5 seconds. 4, 3, 2, 1. So it looks like most of us nailed it. When we look at this, blank, an organ is multiple tissue types working together, and then an organelle is a microscopic structure inside of a cell. Well done. This slide, I remember really it bothered me when I was an undergraduate. I remember being a freshman and a sophomore in college, and I was very binary. It was right or wrong. It was true or false. There was one answer, and it was the correct answer. And that's one of the things I loved about my math and science classes. But human beings are more of a spectrum. And at an undergraduate level, what we teach you in an anatomy physiology class is the most common variation that occurs within a human being. But as you go on, especially if you take a gross anatomy class and you're in the lab setting and you're looking at your cadaver and your buddy's cadavers, you'll notice from cadaver to cadaver to cadaver, or person to person to person, there are little differences. You can see some, like maybe your earlobe is hanging or maybe your earlobe is attached. And just like there are little differences on the outside of our bodies, there are little morphological variations on the inside of our bodies. So in this class, we'll teach you the most common way that it appears in human beings, but I need to give you a big caveat. Human beings are complicated, and you are going to see some crazy stuff as you advance in a healthcare career. Some people don't have specific muscles. For instance, some of us can wiggle our ears. Some of us can't wiggle our ears. Some of us have the ear wiggle muscle. Some of us don't have the ear wiggling muscle. Some people are going to have an atypical number of vertebrae. They have an extra vertebrae, so their vertebrae have fused together when they were in development. Other people may be just missing a vertebrae altogether. Or some people are going to have extra organs, or reversed organs, or abnormally shaped organs. every once in a while, you'll find a human being that has all of the organs of their abdominal, pelvic, and thoracic cavities flip-flop from left to right. And that situation is known as situs inversus. So what we're going to teach you is in the green box. We're going to teach you the most common, the normal morphology in this class for the human beings. But there are other crazy things that occur. Some humans, some people, have their two separate kidneys fused together to make a giant horseshoe. Other individuals will have kidneys up high and kidneys down low. Or if we look at the blood vessels branching from the heart, instead of having this classic 3 blood vessels coming off of the aortic arch, they could have 2 blood vessels or 4 blood vessels coming off of their aortic arch. So this is where the surgeons really earn their money. This is where the medical school and all those years of residency pay off. And this is why they have to spend so much time working in a clinical setting to get used to all these weird morphologies that appear within human beings. Now, we as human beings are alive. And there's a certain threshold you need to meet in order to be alive. There's someone in the hallway. We have an empty seat right up here. You don't have to stand in the hallway. She looked really awkward trying to write while standing. So, as we look at living things, guys, gals, all living things are organized. Within our bodies, we have membranes. Within our cells, we have membranes. We have compartments for our bodies. We're also going to have cells. To be alive, you need to have a cell. A cell, and I'll say it again because this is a key take home. A cell is the smallest living thing. So the empty seat is 3 rows up, 4 seats in. When we look at metabolism, to be alive, you need to have some kind of activity, some kind of chemical reaction occurring. Think of how you eat food, you process food, you defecate, you grow, you make new chemical compounds. Metabolism is the sum total of all chemical reactions in the body. All the chemical reactions in our bodies combined is called our metabolism. And to be alive, we need to have a metabolism. We need to be responsive. We need to be able to sense some kind of a stimulus and then respond to that stimulus. So earlier, about last week, I remember I walked outside, I got hit by the blast of cold air, and I said, oh my gosh, forget this. Closed my door and walked back inside. I sensed the stimulus and I responded to the stimulus. When we look at movement, there needs to be some way to move substances within the organism. And this is kind of a sticking point for students because a lot of students will think of a plant or a tree and they think, oh, well, the trees don't move, plants don't move. And while they may not move fast enough for us to see with our naked eye, within the organism itself, there are substances being moved around. There's intracellular movement or intercellular movement of chemical compounds. To be alive, we need to have homeostasis. And this is a unifying theme of anatomy and physiology. Please circle, underline, star, homeostasis. This is a key term we're going to come back to again and again and again. Homeostasis is maintaining a relatively constant internal environment. So if you think of like our body temperature, we need to maintain an internal body temperature of about 98.6 degrees Fahrenheit. You can have a little bit of fluctuation. You can be a little bit higher or you can be a little bit lower, but it's relatively constant. When we are going to strive to maintain that constant internal environment. Or if we look at blood sugar concentrations, or sodium ion concentrations, or blood pH. There's all these different variables that we need to maintain at relatively constant levels to stay alive. If we lose homeostasis, we typically are going to have what's called a homeostatic imbalance, which is a really hoity-toity or technical way to say that you have a disease or a pathology. When somebody gets sick, they've lost homeostasis. And the loss of homeostasis generally means that you don't feel good. You know, you feel pretty nasty when you lose homeostasis. And if homeostasis is lost by too great of a degree, that's when a patient ceases to live. We strive to maintain that relatively constant internal environment. Something else that we need to stay alive is to be considered alive is the ability to reproduce. Now, this doesn't mean that if you don't have a kid, you're not technically living. We're talking at a species level. We as a species, to be considered living and viable as a species, need to make progeny for the next generation, for the continuation of our species. So this is a big picture view of what we need to be alive. We need to be able to make more of us to continue being alive as a species. And then finally, there's going to be changes over time, and this is known as evolution. There's going to be subtle changes from generation to generation to generation that typically build up over time. So let's spend several slides talking about homeostasis. There's two ways, or two feedback loops that we can use to maintain homeostasis. We have the negative feedback loops and the positive feedback loops. Those negative feedback loops are the most common. There are dozens of negative feedback loops in the human body. To summarize a negative feedback loop, if there's a stimulus, the response is always opposite to the stimulus. If you're becoming dehydrated, you're losing water, the response is you feel thirsty and you drink water and your hydration level goes back up. If you are overheating, you sweat and lower your body temperature. If your blood sugar levels are going down, you get hungry, eat some food, and your blood sugar levels go back up again. The response is opposite to the stimulus. And these negative feedback loops are the cornerstone of maintaining that relatively constant internal environment, the homeostatic balance of our bodies. When we lose homeostasis, we are sick or we die. So how do we work these negative feedback loops? Let's talk about blood pressure. Most of you have probably at some point in your life been lying down, you've been horizontal, you've been relaxing on the sofa, and then you stood up really quickly. And then you went, whoa, as you got really dizzy, had that kind of euphoric feeling. That euphoric feeling was because you didn't have enough blood going to your brain. You stood up too quickly. And your body senses that and responded to maintain homeostasis. So in this example, somebody stood up really quickly after lying down. And then there were some receptor organs, some baroreceptors in this case, that monitored blood pressure and noticed that the blood pressure going to the brain was too low. In response to the blood pressure to the brain being too low, the brain processed that information, sent a signal to the heart, caused the heart to pump harder, and also would send a signal to the arteries and cause the arteries to squeeze on the blood to force more of it to go to the brain. In essence, in response to low blood pressure to the brain, the body temporarily increased blood pressure. That increase in blood pressure causes more blood to flow up to your brain, and then you go from feeling dizzy and woozy, and in a period of a couple seconds, you feel normal and fine again. That's a really quick negative feedback loop where you can get a quick response. Other feedback loops take a longer period of time, particularly if they involve some kind of a hormonal response. Those feedback loops usually take minutes to hours, occasionally days, to get your response. We are going to have, as we look at our classic negative feedback loop, three key components. There's the receptor, the integration center, and then the effector. The receptor is usually going to be some sensory organ associated with the nervous system. The integration sensor is going to be typically the brain. Occasionally, it's going to be the spinal cord. But overwhelmingly, it's going to be our brain. And then the effector is usually going to be some kind of a muscle, smooth muscle, skeletal muscle. Occasionally, the effector is going to be something other than a muscle, but most of our responses are going to involve either a smooth or a skeletal muscle making a change, or they're going to involve some kind of a signal being sent to an endocrine gland in our bodies. So to summarize the slide, we sense something, we make a decision based on the information we receive, and then we respond to maintain homeostasis. So this was a negative feedback loop. Negative feedback loops make me happy as an individual because they maintain balance. The opposite of the negative feedback loop is the positive feedback loop. And these are the ones that make me a little uncomfortable. They kind of scare me a little bit because these positive feedback loops, instead of maintaining the balance, they instead amplify a signal. And the scary thing for me about positive feedback loops is you have an exponential increase in that signal that's being amplified. Classic examples of positive feedback loops include uterine contractions during giving birth. Any woman who's given birth can tell you how during the pre-labor stage the contractions are soft, gentle, they really don't hurt that much, and the closer and closer you get to actually giving birth, the contractions get harder, stronger, and quicker. The signal's being amplified. Other examples of a signal being amplified or a positive feedback loop include electrical activity on the nerve or neurons of our bodies. They're known as action potentials. We also could have blood clotting as another example of a positive feedback loop. Or at the midpoint of the female's monthly cycle, there's a positive feedback loop that causes the amount of estrogen in the woman's bloodstream to skyrocket exponentially. There's a nice quadratic curve to the concentration of estrogen at the midpoint of the menstrual cycle. Those are some examples of positive feedback loops. There are not many positive feedback loops in the human body. Overwhelmingly, we have negative feedback loops. But we do have just a few positive feedback loops because occasionally we need to amplify a signal really quickly. Let's talk about how we can move things. A prerequisite before you came into this class was that you had to take a chemistry course. And you probably learned in your chemistry course, at least I hope you learned, all about concentrations and how if you add more of a chemical to a liquid, it becomes more concentrated, and if you add less of the chemical, it becomes less concentrated. And what we find is that as we have concentration gradients within the human body, chemicals tend to move from an area of high concentration over to an area of low concentration. That's called moving with the gradient. A good example would be if somebody had spray deodorant and they covered their bodies in spray deodorant. There's a high concentration of odor molecules right around their bodies. And those odor molecules tend to move away from them and fill the room. They move from the area of high concentration to the area of low concentration, maybe down the hall or maybe your bedroom. Does anybody use Axe in here anymore, out of curiosity? I remember when I was a freshman in college, they were handing it out for free, aerosol deodorant, and we would tape it open, shake it up, and throw it in someone's dorm room. We call that an Axe bomb. Sometimes gradients move against their concentration gradient. So instead of going from an area of high concentration to low concentration, we're going to move it against the concentration gradient. And we concentrate it. And moving something against that concentration gradient, or uphill against the gradient, is going to require the input of energy. So there's a key thing I need to emphasize for you right now. Moving with the concentration gradient does not require energy. It happens spontaneously. If you spend any amount of time studying thermodynamics, this is called the law of entropy. So going from an area of high concentration to an area of low concentration is spontaneous. It just happens automatically, and it does not require energy. But the reverse doesn't happen spontaneously. Taking something that's dilute and concentrating it, going from a low concentration to a high concentration. That requires energy. It requires the input of energy to move something against its concentration gradient. So in our bodies, if we have a high concentration of glucose outside of the cell, so glucose is right here being represented by the, we'll say, teal hexagons. There's lots of teal hexagon, or lots of glucose outside of the cell. It'll move into the cell with its concentration gradient. Or we can have charged ions, where there's a high concentration out of the cell, a low concentration in the cell, and when the membrane channel opens up, those charged ions, typically sodium ions, will rush into the cell. Or we could have another common concentration gradient is going to be heat. Heat will always move from an area of high concentration to an area of low concentration. So in other words, we move heat around our bodies. Most of our heat will be generated by our skeletal muscle tissues, and then that heat from our skeletal muscle tissues will enter the bloodstream and then be distributed to the rest of our bodies to move that heat around and diffuse it throughout our bodies. So, let's poll the audience. During exercise, and I'm going to add a 60-second timer here. During exercise, someone generates excess heat and the body temperature rises. As a response, blood vessels dilate the skin, warm blood flows closer to the body's surface, and heat is lost. What is this an example of? So before you answer the question, let's dissect the question. Somebody is heating up, and in response to heating up, they cool off. The signal or the response is opposite to the signal that triggered it. Would that be, that's a problem. Let me scroll down here. There we go. Is that negative feedback, positive feedback, dynamic equilibrium, integration control, or set point adjustment? We have about 5 seconds left. If you haven't answered yet, just click an answer. The correct response was negative feedback. And I think we understand negative feedback pretty well based on this feedback from the class. Kudos to you guys. Pat yourselves on the back. And that is it for our presentation on the introduction to human anatomy and physiology. We have just a few minutes left in class. It's not worth starting our next presentation at this point. So we'll stop, we'll be done for today, and we'll pick up tomorrow with body planes and directional terminology. By tomorrow, I mean Friday.